Hi, I'm Leo Smith, and I'm a chemistry major here at Concordia, and I'm a junior.
Hi, I'm Kyle Little, and I'm also a junior, and I am a biology and environmental studies major.
So for COS, we will be presenting a pre-recorded presentation of research that we did in fall 2020 um, during a research seminar with Dr. Jennifer Sweatman in the biology department. So to introduce our problem, we're all familiar with plastic contamination becoming a growing issue due to the increasing amount of plastic waste that is being produced. And these plastics are a problem because they break down into particles known as microplastics that are smaller than 5 millimeters. We did some research and found that terrestrial microplastic contamination is a lot more severe than marine contamination, yet is a lot less studied. So we hope that this study um, fills that gap a little bit. Further research, we found that sludge application to agricultural soils is one of the major sources of microplastic contamination in terrestrial environments, though there are many other sources as well. And then we did more research and found that microplastics have the possibility of transferring up the food chain, either from soil into earthworms or from soil into chickens, though this is just one study and there needs to be more research done on this. We also found that um, plants may not be able to uptake microplastics because microplastics might be too big, whereas plants can uptake nanoplastics just fine. From this research, we concluded that animals might more easily uptake microplastics than plants due to size. And this is how we formed our hypothesis that the small mammals, the small terrestrial mammals that we sampled, would have, or of those, the carnivorous mammals would have more microplastic accumulation in their guts than the herbivorous mammals.
So, second of all, we have our methods here. And first of all, we just want to say that we collected most of our terrestrial mammals from northern Minnesota at certain lab research sites of Concordias and around the area. And after we collected them, we preserved them in a freezer until we were able to collect more data um, on them. So we thawed, skinned, and dissected each of them according to existing protocol that we found in a different research paper. Each gastrointestinal tract was then digested with 10% weight per volume KOH at 40 to 50 degrees Celsius for around 48 hours. Sometimes it would be a little bit more than that. And the volume of KOH was such that the mass of the gastrointestinal tract material was only 10% of the volume in milliliters of KOH. So for our experiment, we in total did 41 different samples, with 5 of them being procedural blanks and 36 of them being samples that were either carnivorous or herbivorous. And these solutions were then filtered using the Wattman 541 hardened ashless filter paper. And later they were, or when we were using the filters, we also used vacuum filtration in order to speed up the process. And we dried them in an oven for 24 hours at 60 degrees Celsius um, before we analyzed each of the filters. The analysis of the filters were done using dissecting microscopes, and all the microplastics that we found were collected in small vials, and we used ethanol to preserve them in the vials before we later analyzed them for color differences, size of microplastics, etc. All of the samples were divided into 2 experimental groups, including the herbivorous group and the carnivorous group, and means and standard errors were also calculated for each of the samples in order to assess contamination. And the blanks were especially analyzed to see if contamination was our issue, or if microplastics were present in the blanks. Rates of microplastic accumulation were then calculated for each sample by dividing the microplastic count by body weight in grams and subtracting average microplastic accumulation in the blanks. And of course, after we analyzed them, if we had negative values, we changed the values to 0, since there is no such thing as negative microplastics. The Kruskal-Wallis test was then used to compare between groups using R and the package dplyr in R.
All right, so once we went through all those methods, we ended up with 41 total samples, 28 of them herbivorous, 8 carnivorous, and 5 blanks. When we analyzed percent prevalence, we found that 57% of the herbivorous samples had microplastics in them, whereas 75% of the carnivorous ones had microplastics in them after we adjusted for contamination. We also calculated the means, and we found that the herbivorous group had 0.0506 microplastics per gram of body weight, whereas the carnivorous group had an average of 0.3176 microplastics per gram of body weight. And when we compared those two groups using the Kruskal-Wallis test, we found a p-value of 0.05978, so just barely statistically not significant. So moving on to figure 1, you can see again the percent prevalence, and the percent prevalence was higher in the carnivorous group, but the sample size was higher in the herbivorous group. And then moving on to figure 2, we have graphed the means here and the standard errors. And so the carnivorous group had a higher mean microplastic density, but also a higher standard error.
Now for the discussion. Although all of our mean numbers of microplastics per gram were higher in the carnivorous group when compared to the herbivorous group, p-values of each showed that even though that was the case, there was no statistical significance in analyzing whether herbivorous groups or carnivorous groups had greater numbers of microplastics in them. No conclusions can be made about the effects of diet on microplastic contamination, and the high levels of contamination in our blanks led us originally to that conclusion. So we had to just subtract the mean microplastic densities of the blanks to obtain our graphs that we are now using in the presentation. As you can see on the right, this is just a photograph of a microplastic through the microscope while analyzing one of our samples.
So our hope from this research is that this study can spur future research on terrestrial microplastic contamination and on trophic transfer of microplastics. We hope that future research can use better anti-contamination measures and a bigger sample size so that the data that they yield is statistically significant. We also think that these future studies should find out a better way to digest the terrestrial animal tissues, because when we digested our terrestrial mammals, we found a lot of insect exoskeletons and wood chips on the filters, which made it more prone to error when we were analyzing. So that means that our basic potassium hydroxide digestion was not able to digest chitin or lignin. So future studies should find out a way to digest these compounds. We also think that future studies should use spectroscopic methods in their analysis of microplastics to determine the types of polymers of microplastics that are found in order to determine their source. And such methods could include FTIR or Raman spectroscopy, or many others. So to conclude, microplastic contamination in terrestrial environments is very understudied. And so our hope is that this study can inspire more studies on terrestrial microplastic contamination and trophic transfer of microplastics. And hopefully we can gain some important insights about those topics.
Thank you for coming to our presentation. And if you have any more questions, feel free to come to our Q&A session on April 14th at COS. Thank you, everyone.